Did you know that a lack of sleep can cause parts of the brain to be eaten or consumed by other brain cells? But before we go into this, let me explain the different types of brain cells. There are two main groups: the neurons, which are the nerve cells that send and receive signals. The site of two of these cells joining is called a synapse. The second group is glial cells, which are the supporting cells of the brain. One type is an astrocyte, which regulate the transmission of electrical information. There are also microglial cells, which remove damaged neurons to maintain brain health. The microglial cells clear out old and worn-out cells through a process known as phagocytosis. This process allows a cell to use its plasma membrane to engulf a large particle, essentially consuming the other cell. Our brain changes states when we sleep to clear away the toxic byproducts of neural activity experienced during the day. However, the same process occurs in individuals who are chronically sleep deprived, but the process becomes more agitated. Microglial cells and astrocytes become more active in a brain that is experiencing chronic sleep loss. Instead of the microglial cells only clearing away old and damaged cells, they begin to consume healthy and important neuron cells. This is a concern, as it has been observed that the likelihood of obtaining neurodegenerative diseases such as Alzheimer's increase significantly. This topic is further explored in a study conducted in 2017. Michael Bellesi and his team studied the brains of 4 different groups of mice. The first group were left to sleep for 6 to 8 hours, which is the recommended amount of sleep. The second group, or the spontaneously awake group, were periodically awakened from sleep. The third group had mice who were acutely sleep deprived, who were kept awake for an extra 8 hours. And the final fourth group were kept awake for 5 days straight to induce chronic sleep deprivation. The study showed that particular cells, such as glial cells, were more active in the mice who had been acutely and chronically deprived of sleep. Instead of just the microglial cells eating waste, which is known as astrocytic phagocytosis, the astrocytes had increased their activity and were actually consuming parts of the synapses. Essentially, the brain goes overboard with clearing and starts to harm itself instead. Here is an example of a brain cell, which is in green, being consumed by a glial cell, which is in blue. An example of a signaling pathway which recognizes these eat me signals is MERTK. It has long-term negative consequences on the brain structure, and this was seen in the study in the mice who underwent sustained sleep loss. This structural damage is due to translocated phospholipids, which cause increased calcium concentrations and ATP becoming depleted, ultimately triggering the brain to consume itself.